Ja, herzlich willkommen zurück hier ins HORSCH
So,
Live-Studio zu
welcome
unserem
back
zweiten Tag bei
in the
HORSCH
HORSCH
Live.
Live Studio.
Für
It's
alle,
the
die
second day of
neu
HORSCH
dazugekommen
Live.
sind,
All
wir
that
haben
just
an diesem
joined
Tag
in.
Vorträge im Bereich Düngung,
Today we are
Nährstoffeffizienz
having
und das Ganze auch in
talks
4
about the
Sprachen. Das heißt,
efficiency of
wir können die
fertilization.
Inhalte
We
in
do it
Deutsch,
in 4
Englisch,
different
Russisch und
languages. That
Französisch
means we do
anbieten.
it in
Ja,
German,
wie in
English,
unserem Vortrag
Russian, and
von gerade eben,
French.
wo wir den Stickstoffmarkt oder
As we
generell
already
den Düngermarkt
heard,
beleuchtet
when we're
haben,
talking about
wollen wir jetzt natürlich
nitrogen and we were
eine
talking
Etage
about the
tiefer
nitrogen
gehen,
or
im wahrsten Sinne
fertilizer
des Wortes.
market,
Wir
we
wollen
would
in
like
den Boden
to
reinschauen
step just
und
one
aus einer
stair deeper
wissenschaftlichen
and have a
Perspektive uns
look
anschauen, was wir denn
into
tun
the
können,
soil
dass wir ein
from a
möglichst effizientes
scientific point of view,
Wurzelsystem
what we
haben, dass wir
can do in
Nährstoffe
order to
möglichst
have a
effizient
very
aufnehmen können.
efficient
Und
root
das ist sicher
system
eine
that
Mischung aus
can take the
einmal der
fertilizers
Pflanze
up
an sich,
very
aus sicher auch
easily and the best
dem
way
einzelnen
for the
Genotyp an
plant. And
sich,
this is
aber natürlich
very
auch die Frage, mit
interesting for
welchen
the plant
Nährstoffen
itself,
kommen wir
and
daher,
depending
wo
on the
legen wir
genotype, of
Nährstoffangebote
course,
im
we always
Boden
have to
hin
talk where we have to
und
put
wie können wir
the
das Ganze
fertilizers, which
letztendlich
fertilizers are to
optimieren.
be used,
Wir haben uns dazu als
and how can we
Referenten Herrn
optimize
Professor
that.
Dr. Nicolaus von
As a
Wirén eingeladen. Herr von
speaker,
Wirén ist
we invited
Institutsleiter
Dr.
für Physiologie und
Nicolaus von
Feldbiologie
Wirén.
am
He is the
IPK in
head of the Department
Gatersleben.
of
Seine
Physiology and
Abteilung
Cell
beschäftigt sich
Biology
vor
at
allem
the
damit,
Leibniz
wie
Institute of
Pflanzen
Plants
auf
in
unterschiedliche
Gatersleben.
Stressfaktoren
He's
reagieren und da
actually
in
talking about
Besonderheit
plants react on
auf
different stress
Trockenheit und
factors,
Nährstoffmangel oder
especially on
andersrum
drought
formuliert
and
natürlich auch,
lack of
wie man mit
nutrients,
bestimmten
or the other way around. You
Nährstoffarten
can also
eben
say that
Wurzelsysteme
how you
optimieren
can
kann. Die
improve and
Erkenntnisse aus der
optimize
Forschung
root
gehen natürlich
systems
auch
by
rein, um zu
aborting
sagen,
good
wie können wir in der
nutrients.
Züchtung
Of
weiterkommen
course, you
oder
know
jetzt
him,
in
and
unserem
we
Fall,
also
wie
talk
können wir
about
denn
how can we
technisch
do
auch
breeding
Nährstoffe
differently,
in
or how
Bereiche des
can
Bodens bringen,
we
wo sie
uh,
sinnvoll angelegt sind.
put
Ja,
the
wir haben den
nutrients
Vortrag
in
im
certain
Vorfeld
areas
aufgezeichnet, aber wir
of
werden
the
natürlich
soil.
im Anschluss
We
daran eine
recorded the
Live-Diskussion mit Herrn
presentation
von Wirén
beforehand, but
haben, wo einfach
after
Ihre
that, we
Fragen,
will
die
have a
im Chat
live
kommen,
discussion
beantworten zu
with
können.
Nicolaus von
Ich fordere jetzt
Wirén
auf
in
einen
order
spannenden
to
Vortrag.
answer your questions. I'm looking forward to a very exciting presentation.
Einen schönen guten Tag. Mein Name ist Nicolaus von Wirén. Ich
Good
bin hier Leiter
morning.
der Abteilung
My name is
Physiologie
Nicolaus
und
von Wirén. I'm
Zellbiologie
head of the
am
Department of
Leibniz-Institut für
Physiology and Cell
Pflanzenernährung und
Biology at the Leibniz
Kulturpflanzenforschung
Institute
hier
of
in
Plants,
Gatersleben.
Genetics and
Meine
Crops
Arbeitsgruppe
Research here
arbeitet in der
in
molekularen
Gatersleben. My
Pflanzenernährung,
work
aber
group in
wir
molecular plant
interessieren uns
nutrition,
auch
but we
insbesondere für
also
die
are
Beziehung
very
zwischen der Wurzel
interested
und den
in the
Nährstoffen.
relationship
Und
between the
genau
root
darüber
and the
möchte ich mit Ihnen
nutrients.
heute
And
auch
this is
sprechen. Es
exactly
geht
what
also
I
um die
would like
Anpassung
to
von
talk
Wurzeln
to you
an
about
äußere
today.
und innere
So it's about the
Nutrienverhältnisse.
adaptation of roots to the
Wenn
external and
wir uns jetzt hier
internal nutrient
zwei
conditions.
Sommerweizenlinien
If we
angucken,
have
die
a
beide
look
übrigens aus
to
der
two
gleichen
summer
Kreuzung
wheat
stammen, dann
lines,
sehen
both
wir,
of
dass die
which, by the
sich
way, come
nicht
from the
nur am
same
Strauch
crossing,
unterscheiden,
then we can
sondern auch an
see
der
that
Wurzel.
they do
Und
not only
zwar hat
differ in
die
the
linke
shoot,
Pflanze
but
hier
also in the
zwar
root.
deutlich mehr
At the left,
Wurzelbiomasse, aber die
you
rechte
can
Pflanze,
see
die
much
hat
more
ein
root
längeres
biomass, but
Wurzelsystem.
on the
Das
right-hand
heißt, wir
side,
müssen
we
uns
have a
hier
longer
nicht
root
nur die
system.
Biomasse
That
angucken,
means
sondern wir
is
müssen
not
uns
only
unsere
enough to look at the
Architektur
biomass, we
anschauen.
also have to look at the root
Diese
architecture.
Architektur, die beschreibt die
And
räumliche
the root
Anordnung von
architecture
Wurzeln
describes the
unterschiedlicher
spatial
Ordnung.
arrangement
Und diese
of roots
Wurzelarchitektur
of
hängt zum
different
einen vom
order.
Genotyp
And
ab.
this root
Der
architecture
Genotyp ist also
depends on the
letztlich die
genotype.
Sorte,
So
die Sie
the
auf dem Feld
genotype
haben. Und der
ultimately is the
Genotyp bestimmt
variety
sozusagen
you
den
have in the
Aufbau,
field,
den
and the
Bauplan
genotype
so einer Wurzelarchitektur.
determines, so to speak, the
Auf der anderen
blueprint
Seite
of the
ist die
root architecture.
Wurzelarchitektur auch abhängig von
On
den
the other hand, the
Umweltfaktoren,
root
die sich
architecture
bisher
is
gezeigt
also
haben,
dependent
von
on environmental
einer
factors. And here you
weltweiten
can
Pflanze.
see
Hier auf
that
der linken Seite
we
sehen
have
wir
a
Rüben,
two and
das
a
sind
half
ohne
year old
Anweisung, in Abwesenheit
plant.
von
The one on
Mikroorganismen
the left side is
angezogen
sterile, so
und hier auf
without
der
instruction of
rechten
the
Seite
absence
in
of
Anwesenheit von
microorganisms. On
einem
the right
rhizosphären
hand side,
Bakterium. Das heißt,
it
bei uns
was
ist das
done in the
hier
presence of a
das
Rhizosphere
Silberbakterium, welches viele
bacterium.
Bakterien zu
In
deutlich
our
längeren
case, it means the
Zeitwurzeln,
plate.
vor
And here
allem
you
in
can
den
see that
unteren
the
Wurzelblättern führen.
bacterium leads to
So,
much
das heißt
longer
also,
times
diese
roots,
Umweltfaktoren wie zum
especially in the
Beispiel
lower root area.
Bodenmikroorganismen,
That is, that
interagieren
environmental
auch
factors
mit dem
such as
Genotyp
soil
der Pflanze.
microorganisms also
Und
interact with the genotype
diese
of the
Interaktion
plant.
ist auch nicht
And
stabil,
this
sondern
interaction
verändert sich
is
im
not
Laufe der
stable,
Pflanzenentwicklung,
but it
sodass wir
changes in
letztendlich
the
von
course of the
einer
plant development, so that
Wurzelplasticität sprechen.
ultimately we
Die
speak
beschreibt
of
die
a root
dynamischen Veränderungen
plasticity,
der
which
Struktur, der
describes the dynamic
dreidimensionalen
changes in
Verteilung
structure,
und des
the three-dimensional
Ausbreitungsgrades von den
distribution,
Wurzelsystemen.
and the branching degree of the root
Schauen wir uns
system.
jetzt mal das Wurzelsystem von
So let's
Deficiency
take a
etwas genauer an.
closer look to the root system of barley. The barley during germination forms so-called semi-serous roots. That means they originate from the embryonal state and usually are 4 to 8 in number. 2 to 3 weeks later, the barley still forms normal roots, which originate at the stem base of the head and from the sides. And if we look at them and look and see the comparison, we can see that in the second picture that the synodal roots are slightly larger. And in the microscope, we can also observe that they have a larger number of light vessels, of cool vessels in the central cylinder. And this larger diameter of these vessels also leads to the fact that water and the nutrient transport ratio over this type of roots is larger. So, in the course of development, these seminars roots do not grow at the same time. We can see plants on day 18th, which is actually only the seminal root system here at the left side, and then with a distance of 1 week, we see that the nodal root system comes along. And if we have a look at the biomass and the length of the seminal roots, we can observe that until day 39 and day 36, at least here in the hydroponic culture, where we grew the barley, and the growth of the roots stops earlier, let's say already at day 32. But the nodal roots arise much later, let's say after 2 or 3 weeks, but then they increase exponentially in their biomass. And at that moment where the seminal roots decrease, means they don't grow any longer. That means that seminal roots form the early root zones in the nodal root system, the later root system. As we already seen before, the synodal roots, which come later, can arise at the base of the stem, but not from the main shot, but also from side shots, which can be shown in this picture. We had a closer look to this, and in the crossbreed that we did together with Strobel research, namely from a genotype that was weakly stocked, like for example a single genotype, and then a genotype with a long stock. And here we have several crossbreed offsprings, which also can increase from left to the right. And this is what you can see. We also counted it. We only had 1 to 2 shots, and here we have around 7 in this middle genotype. What you also can observe is that the root length already increases significantly, so that actually it looks as if a genotype with many with many extremes has a higher root depth. But I have to say that we have seen that only in this crossing and not in other intercrossings. So this is something that we will have to explore even further. But what was interesting for us, it was the number of the nodal roots that actually occur at the side shots. And therefore, this is why the upper line of this 6 lines, we have the main shot with the side shots and their modal roots. What we can observe here is that the modal roots still can be formed up to the 7th main shot. That means that new nodal roots will grow always. That means that there is a higher number of nodal plants, and this correlates very strongly, as you can see, for example, here the right picture below. There's a very good safety, there's very good probability for that. So there is a big difference of genotypical variation of formation of shots and also of nodal roots. The nodal roots also have to be thought of. If we now look at different barley genotypes that we've grown here in our root boxes, these are boxes for root observation or roots of thrones. So if you really see them, that they grow inclined, then the roots grow along that plexiglass screen, and you can take nice photos of that. And here you can see even yet another difference in the root architecture, meaning that the genotypes can difference in the root angle. What we can see with the genotype 1 is a relative shallow root angle. Here in genotype 2 it's much steeper. And we can also see that if the angle is steeper, the root depth is also slightly larger. So why should it make sense that I distinguish plants in their root angles? This is something that we found out on the basis of studies of the different phosphate efficiency of soybeans. And here we can see a picture of a colleague from the United States. So they're efficient soy lines compared to inefficient soy lines on the ground where the phosphate was very strong fixed. And just as a reminder, phosphate is usually found in the upper line of the soil because it rises from the generation of organic matter or it is fertilized, and therefore it does not shift into the depth because phosphate precipitates just very easily, whereas nitrogen, of course, is also generalized from the organic matter, but then also after quite shift deep the soil layers. And if you have a look again at those efficient phosphorus soybean genotypes, in their root types, you found that the phosphate efficiency is actually forming a shallow root systems, but the ones that are more inefficient actually have a deeper going root system. That means that if the phosphorus is localized up here, it's quite clear that a larger root surface area can absorb more phosphate. And this can also be observed in the intake of the shot, which is significantly larger in these genotypes than in the other ones. So shallow root system is good for the efficiency. So in general, the plants usually react very strongly to localized nutrient offers. And we can also observe this here, this attempt that has been very, very old, stemming back in 1975, but actually has a very strong value for demonstration. Here, individual seminal woods of barley were exposed to a placed nutrient offer so that, for example, only phosphate was offered here in this central zone, whereas the upper parts of the root and the upper part of the lower roots grew under phosphate deficiency. And here you can see clearly that under this local phosphate offer, there is leads to a strengthening of the side root growth, so more and longer side roots than here the other side, where the phosphate was offered evenly to all root zones. If you do that with nitrates, so local nitrate offer, it looks actually quite similar, even more and longer side roots, also where this nitrate supply can be found. In the case of anomium, it's a little bit different. Here we can see significantly more side roots, but these side roots are also much shorter. And what I consider to be very interesting, this is an observation that does not apply equally to all the nutrients. If we make a local potassium offer, and here there is no potassium, at the top and at the bottom, we do not see a stronger reaction. So there is no enlargement of the growth, but actually, this is what it looks like in the culture. That means that in the course of their evolution, the plants have not adapted to any nutrient supply with increased root growth, but only do it with existing ones. And it's particularly pronounced in the case of phosphate, nitrate, and as we've also seen in the case of ammonium. And these differences between ammonium and nitrate can also be illustrated a little bit better. If now you have a test system with small plants, so like the field baffle, here in a divided plate where we have the main root in the left part, which is weakly supplied with nitrogen, but then we have a placed and localized offer of nitrogen on the right side. And there, the first side root is Wirén, and we can see reaction on the nitrogen supply in the form of nitrate with a significantly longer growth of the first three side roots of first order, and also with a significant extension of the side roots of second degree. So let's make the same drawing with the localized offer of ammonium. Then we can see that the growth is much shorter, and the side roots of second degree are also much shorter. But we can also see that there is a strong, more pronounced branching. So there are side roots of third degree. So in short, ammonium increases the branching of the side roots. Nitrate, on the other hand, is very positive for the extension of the roots in the local supply. And you can also see that if, for example, in this case, you can only see corn plants converted from an ammonium nitrate mixed diet to a nitrate for 4 days. This is what we can see. With nitrate, we can see the long side roots, and with ammonium, they're significantly shorter. So why is that so relevant for the farmer? Also, if you think of local ammonium depot, for example, the ammonium phosphate, but here it was only done with ammonium, also to have it in a root box. And so we can see that the ammonium fertilizer can instruct. And as we can see here, wheat roots slowly approach that. First, they stay at a distance because ammonium, the first phase, still is in its old form, actually is toxical if the concentration is higher. So the root wants to prevent it. But little by little, it grows into the direction of the ammonium fertilizer because the ammonium nitrate affects the ammonium concentrate. And what is also important, what I would like to say, and you can see it in this middle picture very well, here it is in the vicinity of the ammonium also is a strong branching of the right side roots. And at the later moment, the growth rate towards the ammonium depot is also significantly larger than the side root growth rate is in places that are not supplied with nitrogen or with little nitrogen. So that was also here in a Chinese work. It was also captured with a photo. Here you have fertilizer ammonium together with phosphate, and here you place it nitrate together with the phosphate, and you can see that the root formation was not so strong. So good. So far to the root architecture and its influence by local nutrient supply. But it is also interesting that the state of nutrition of the plant has an important influence on the root formation. And this is what we can see here with bean plants that have been grown in close nutrient solution that's very efficient, or with the deficiency of potassium, magnesium, or phosphate. And as you can see, with potassium and magnesium deficiency, the shoot already grows quite nicely, but runs very strong in the plants. Here we can see a large one with that leaf close, which then after passes into a large one as the potassium deficiency, and there is here, and we can see that it really affects strongly the root growth. So a deficiency of potassium and magnesium, the root grows in a very limited way, whereas in the case of deficiency of phosphate, the shot will suffer. But of course, the root growth is much stronger. And this depends on the distribution of assimilates. So like the sugars from photosynthesis, whether they can still be transported to the root or not. And this is something that was confirmed here by sugar analysis. Here we can see the percentage of sugar in the control group, 44% still in the shoot, 16% were shifted to the root. But in the case of potassium and and magnesium deficiency, hardly sugar can be found in shot. So this is connected to the fact that potassium and magnesium are important for the assimilate displacement into the roots. In the case of phosphate deficiency, the reaction is just the other way around. And therefore we have this strong root growth. So the state of nutrition already has a very large influence on the biomass of a plant. Then we can also see, under nutrient deficiency, for example, there is an increased formation of root hair. This can be shown here in the comparison between a plant of deficiency with iron or phosphate, and we can see in the case of lack of iron, there's an increase an increase of root hair. Here we can see from 33 to 45 root hairs per millimeter of root. Under phosphate deficiency, it's even more, up to 65 root hairs have been conducted here per millimeter. And these root hairs are also much longer. So what is actually very interesting for us is the reaction from the roots and the side roots to nitrogen deficiency, because we have to recall that with the amendments of the fertilizer regulation, of course, we're allowed to use less nitrogen in our fertilizers, and that also means that the plants are very often in a slight situation of shortage. So how does the root react? With this slight onset of nitrogen deficiency, it reacts on the one hand side with an increased number of side roots, and these side roots are even longer. This also means that this is ultimately a reaction of the plant with which the plant tries to increase the volume of the soil from which it can draw the nitrogen. So this reaction can be seen very well if you see here our model plant field baffle, in which we work a lot because it's so well characterized molecularly and genetically. So if you think them, if you grow them with a very low nitrogen supply, you can see that with a mild deficiency, it comes to an extension of the side roots. And if the nitrogen deficiency becomes even stronger, the root growth will reduce. And this is a very first reaction, the mild nitrogen deficiency, what interested us particularly. We wanted to know the genetic reason behind it. So, and now I'll show you an example. That's why we researched 200 different lines, that means genotypes, and we put it either to a good nitrogen supply or planted it into soil with little nitrogen supply, so that the plant should suffer from deficiency. And here I show you an excerpt of the root system of different lines, here with high and low nitrogen offer. And you can see here in the far left, there's a very weak reaction of this genotype to the nitrogen deficiency. But on the right side, we can see that these genotypes react very strongly to the deficiency of nitrogen with a prolonging of the side roots. So there is a very big difference between the different genotypes, and we want to find that out because, of course, we want to bring such genotypes in the field which are able to get the nutrition even if the nitrogen is far away from them. There is a procedure for this. If you have such different genotypes that show different phenotypes, that means show root lengths, then you can associate these characteristics with genetic markers, that means associate them, and then map the genes that are responsible for that, for example, for the longer root growth. And you can describe that in a so-called Manhattan plot, which then shows which marker reacts particularly strongly to this characteristic of root lengthening under nitrogen deficiency. And so we found genetic variants, which we tested with the help of genetic transfer, that means genetic manipulation. And for here we have two examples. Here we have the wild type and the mutant, which no longer carries the gene at all, and we have the gene variant 1 and gene variant 2. These genetic variants do not lead to any clear difference if the plants are correctly supplied with nitrogen. But if the plants receive very weak nitrogen supply and have a multi-differency, we see the extension of the side roots and primarily. But in the genetic variant 2, we see that the side roots even are 15 to 20% longer than in the genetic variant 1. This is something that we did not only find for this gene, which is called BSK3, but we also found it for another one, which is called UCA8. And also with this gene, we only see, or with this variant, we only see under nitrogen deficiency that the extension of the side roots is much longer than in the case of the genetic variant 2. Actually, I do not want to make it too complicated, but I would like to say that these genes play a very important role in the synthesis and transmission of hormonal signals of these hormones called steroids and also endoxines, which are both needed that the plant can form the prolongation of the roots under nitrogen deficiency. And with these two genes, of course, we now have markers which allow us to try to transfer it to cultivated plants, let's say to transfer the knowledge to the cultivated plants. And what is really interesting about these genetic variants is that we only have a change from one of the base, that means of one letter between billions of letters that we find in the genetic code of a plant. And here in this YOC18-2, so that corresponds to 2 letters within the DNA, or 2 digits of the DNA. Only if we change these 2, we can already achieve much longer roots. And now we tried to find these genetic varieties in our genetic bank. But interesting, we will not find the same variant there. But for example, for the BSH3 gen, we only find the weak variant. That means that here we cannot proceed, cannot get any results with breeding and crossing. We need another instrument, which is the genome editing. That means that now we can change 1 or 2 letters with a surgical intervention in the DNA, and with this we hope to achieve longer side roots under nitrogen deficiency in barley. Okay, so now I would like to explain with 1 or 2 slides what we actually do at the EBG. The Leibniz Institute in Gatersleben has the largest genetic bank in the EU, with seeds of more than 3,000 cultivated plant species and a number of more than 150,000 lines that are of defined geographical origin. And we have a special focus on barley and wheat, and we're now trying to preserve these seeds that we have more. That means that they are grown every 15 to 20 years and of course properly stored and in the dry conditions and at minus 18 degrees. But we also try to characterize the genetic constitution of these lines with the genetic markers, and now put these markers or data and also phenotypical data such as flowering times into the grid, or into the we put them online. So we have a digitalized genetic bank, which represents a kind of resource center for the genetic diversity of cultivated plants. And as already said, we're now trying more and more to sequence those barley and wheat lines, and of course we develop new breeding technologies or marking for the genome sequences that can be used for breeding, and try to combine them to improve the genetic constitution. That's what's called a phenotype. So if you record the quantitatively shape and physiological performance of plants. And I would like to show you a small film now, which I brought for you. So, here we see the EPK. This is our site. We have about 10 hectares of land here and 90 hectares for field trials. This is the plant culture hall, which has been newly built. And actually, it's unique in the world. This plant culture hall has 2 different cabins. In these cabins is where the fertilization is taking place. On one hand side, directed at the root. You can see that here there are such mobile towers which move between the rows. In these rows, we see the Rhizosphere. So these are root showcases in which we grow the plants, and these root showcases are introduced totally automatically into those towers, where they are scanned and where photos are taken. So finally you can get an image of the roots just as they develop under the plexiglass screen. And in the second cabin we have a plant cultivation in containers. The principle is that the sensor gets towards the plant, meaning that up here there is such a crane and with cameras which film and take photos of the cameras. Here we have a comparison of the plants growing in the field, and here how they look like in the container. And in this situation we can simulate the environmental conditions that exist outside, and you can imagine that this is a very, very important tool for the future, because now we are dealing every time more and more with climatic changes. So it's really important that such climatic changes can be simulated in order to see how the individual genotypes behave and react to these changed climatic conditions. Well, in these containers, now you can see how they are cut open. There are highly controlled growing conditions in the soil. That means that the soil moisture is measured and controlled by sensors, also the soil temperature. There is still vertically introduced tubes where camera systems are installed so we can observe the root growth. Okay. So that was this. Talking about the phenotypization. So, but now can we take this research results to the field? And at least in my work group, we further developed another method, or tightened it up, like using a postdoc mounted on a tractor. This is for our help, and this allows us to gain a soil profile of about 1 meter in length. This the content is wrapped or packed in a bag, and when we open it up and cut it as if it were salami, from each slice we can extract and recognize the DNA. And in parallel we have developed a method that allows us to recognize specific DNA from an individual plant, so that we can really quantify the root mass of the plant in the segment of soil. So the result is practically a kind of illustration. We did that here with 4 root species, which is the mustard, Celia, and Alexandria clova. And now we can see in this figure that the biomass and the root biomass, but the root biomass is now divided into individual soil layers. And then we can see how much root biomass can be found in the depth of more than 70 centimeters. And here we can also see that oats have a lot of biomass, especially in the upper part of the soil, whereas the Phacelia has most biomass in the medium soil. And the Alexandrian clover has formed much less deep biomass or less biomass at all. Then we have a mixed culture where all four fruits were proportionally represented, and we can see that the total root biomass also increased a little bit. This means that these fruits differ very clearly in the formation of root biomass, but also they have a big difference in depth of the root formation. So has this also an effect on the intake of nutrient, which is something that we also investigated with our colleagues in Hanover, who determined the nitrate and phosphate on at least 3 different depths of soil. And we can see in the case of mustard, which reaches also the lower layers of the soil, that there is a significant depletion of the nitrogen boom. There is no so much change in the phosphates, and in the oats also we can see the same thing, but in the Alexandrian clover with its root depth, which does not reach the soil layers, we cannot see so much difference. So actually we can see that the absorption efficiency of nitrogen from lower soil depth is also related to the root biomass that is formed in the different depth of the soil, and there it really differs clearly between one fruit species and the other. So the question is, what influence has this morphology of root or the significance of the root for the nitrogen transfer for the following fruit? Here we can see the quantity of nitrogen that was in the shot, and on the lower side we can see the amount of nitrogen that has been fixed in the root biomass. We looked at that in 3 different locations, so let's say 3 different environments, and we can see that, for example, here, this year that was Ottendorf, in 2015 it was about 60 kilograms, and that Phacelia and mustard and oats had with the sprout biomass, whereas the root biomass here in the case of mustard was 45 kilos, in the Phacelia it was over 200, and in oats it was 100. So it's actually the much more from the transfer of the shot, but that hasn't always been to be the case, because in Altendorf in 2016 we had it was a dry year with very nutrient-rich conditions. That means we had little root formation, but in Troisdorf in 2016 it was also a very dry year in poorer nutrient conditions, and here we can see a very little transfer via the shot, only let's say about 20 kilos, but through the root it was much more, it was between 15 and 60. And again here the best results with the mixture of different grains. So that means that the transfer to the sequent grain can be greater than of the biomass, which is above there, and the mixed culture was always the same, or at least as good, if not even better than Ukraine. So that means that we also looked at how the following fruit develops after the between. So corn was our following fruit. We did not only determine the yield of corn, but also we were interested in seeing how the root growth reacts on that. And this is what we can see here on the basis of the root, the fresh mass of root in different soil layers. This is corn after mustard, after Phacelia, and oats and Alexandrian clover. And what is really amazing is that that in the case of mustard, we see that the root growth was a little bit inhibited, not so deep, like, for example, with the fallow land. But to the contrary, in the case of Alexandrian clover, the root growth of maize went much, much lower. And this effect cannot be explained by the fact that the roots of the former fruit physically had prepared the soil. Actually, it should be the fact that not a physical effect is taking place, but rather a chemical or biological effect. And something like that, of course, has to be further investigated, but we can see a clear, a very good effect of clover to the formation of roots in the case of corn. And this takes me to the last chapter that I would like to discuss with you, namely the physiological performance of the roots. The roots are, just imagine, are small biochemical power plants which can change a little bit the location of the site prosperities, at least in the roots area. And here's an example of a cucumber plants that were grown in nutrient solution here with good or bad iron supply, and we took it into the field where we have added the pH indicator, which turns yellow when the pH drops. This is what we can see here, that the plants living under iron deficiency significantly lower the pH in the atmosphere. So we have more proteins in the rhizosphere. Okay, this is something you might already know. Like, for example, in the case of wheat plants, if you offer them different forms of nitrogen in the form of nitrate, here on the right-hand side, it's ammonium. Then again, a layer over the soil, and with this indicator, you can see a purple coloration in the case of nitrate. That means that the level of nitrate increased near the roots, but in the case of ammonium, it's an acidification. We talk about a physiological acidification of the rhizosphere. And this is physiological acidification, as you can see, also reaches a few millimeters into the soil. So this is the spatial expansion of the rhizosphere. And in this rhizosphere, we get an increased pH value of poorly soluble nutrients such as phosphate, iron, zinc, manganese. But the plant can not only change the pH value in the rhizosphere, but it can also lead to chemical reactions. And this is something that we can also show by plants living under iron deficiency, which we have the ability to reduce three-finished ions in divalent iron in color. And this divalent iron can be fixed with the help of a complex builder, and then it turns red. This is what you can see here. So the plants living under iron deficiency can also, and you can see that in the rhizosphere, especially here near the roots, tips can reduce free-finished iron to divalent iron. That means that chemical processes also take place in the rhizosphere. And it even goes a step further, because these processes, which I've just shown, play a direct role at the root surface, but the plant also gives up exudates. And these root exudates can be, for example, fluorescent, as you can see in this example, because here we have wild plants that have grown under conditions of iron deficiency, and we simply irradiated it with ultraviolet light. And you can see that the roots start to fluorescent, and here is a mutant which can no longer form this certain root position, and this is not the case. But if you take those fluorescenting blades and you took them out of the field, you can see that some of the fluorescence remains in the former medium. So that means that the plants have released fluorescent compounds. And a few years ago, we also managed to identify these fluorescent compounds. They are so-called coumarins, and these coumarins are released by leaves of two-leaf plants, and this is made as soon as they feel iron deficiency. And this starts relatively early, meaning with a pH of 6, 6.5 towards 7. Well, these processes already take place. So that means coumarins are released via the roots, and these coumarins are able to reduce iron and at the same time bind it into a complex. And then they bring the whole complex back to the root where the iron is absorbed. Well, these root extension can also be very important for the nitrogen upload of the plant. And this is something that we could demonstrate together with colleagues of the University of Bonn, who also mainly carried out this study and investigation. They identified different corn mutants and identified a mutant that was particularly sensitive to nitrogen deficiency. That means that there was very long of chlorine and reacted immediately to a reduction of the nitrogen supply. And interestingly, this mutant is known to no longer form flavones, and these flavones are a certain group of secondary metabolites which plants actually form. And they made an interesting attempt, meaning that they exchanged the Rhizosphere, that is the root-influenced soil between the wild type and the mutant. And if you exchange that, meaning that the mutant is grown on its own soil, that's unadapted soil, so to speak, where you can see the deficiency of nitrogen here, again, the central example. But if you transfer the mutant to the soil from the wild type plant that can form this zone, then the nitrogen deficiency goes back. That means that the nitrogen deficiency can be remedied. And in order to show that this has to do with the soil microorganisms. Microorganisms were genetically identified, means sequenced, the DNA was sequenced, and we had different bacteria families which were identified and appear in this diagram. We can only see that the composition of the soil microorganism is significantly different. Each color stands for a different family of soil microorganisms, and we can see in the wild type there is a lot of blue, and that has decreased significantly with the mutant, which no longer forms the flavones and no longer releases them through the roots into the soil. And ultimately, the dissolution or the current model of this mechanism is that corn plants, if they can form flavones, expede the flavones with their roots and have a strong influence of the composition of the soil microorganism, as we can saw in this case. A very special family is promoted. These are the oxalobacteria, and these oxalobacteria are in the shape to form own substances, which are hormone-like substances that lead to increased root growth and also improve the nitrogen absorption of the plant. So this should show once again how important it is that we actually think about the physiological processes at the root when we expose plants to non-abundant supply of nutrients. So, and finally, I would once again give you an example of how important it is that plants are not only thought with their world of microorganisms, but also together with other plants, because this is how they were created in the course of the evolution. And that's why I now come to, I would like to talk about the mixture of culture. Here's a study of China, which lasted about 15 years, where maize was grown either in a mixed culture, which is the blue line, or in a pure culture. and then the yield was met. And here we had a mixture either with rapeseed or with beans. And what we can see here, there is not a clear deviation, but you can see a tendency. That means that the development of the yield of the corn was much better if it was in mixed culture. Of course, you will say, well, mixed culture is very difficult, etc. And of course, this is very clear to me. But just let's think that all those microorganisms that live in the soil are important, and they are much more multifold in a mixed culture. And this, of course, can be found in the different plants. And this is an effect that will increase with the time. Well, just to come to an end, I would like that genotypes and roots are really different in various things. The architecture of a root refers very much of the type of amount of fertilization, and we have seen that with a slight lack of nitrogen, the roots become longer. With local ammonium offer, many side roots are formed, but they're relatively short. With local nitrate or phosphate offer, the roots get longer. Then we also saw that the combination of between fruits with different root properties can ultimately increase the uptake and transfer of nitrogen to the following fruit. And we also saw that certain root depositions can improve the composition of microorganisms in the soil. So they can improve the microbiome in the soil and ultimately promote the growth of the plants. So this is also the basis of the positive effect of the fruits or mixed culture. And after all, genotypes and varieties differ in many important root characteristics. That means that the breeding improvement of root traits should increasingly come into focus. And these improvements can benefit clearly from molecular breeding measurement methods such as gene editing. This is the genetic scissor we were talking about, and where last year a Nobel Prize was done. Well, and at the end, I would just like to thank my group, which always supports me very strongly, because they don't only support me, but ultimately carry those studies out. And of course, I would like to mention the funding for research from the Federal Ministry of Education and Research, also from the Ministry of Food and Agriculture. I've shown here an example of the cooperation between the breeders and strawberry research. Of course, we receive our main funding from the Leibniz Association, and then again and again of individual projects of the German Research Foundation. Well, with this I come to an end, and I hope that I could awaken your interest in this unknown half of the plant growth in the soil, which is the roots, and could strengthen it a little bit.
Herr Dr. von Wirén, vielen Dank für den interessanten Vortrag. Ich denke,
Dr. von Wirén,
es ist ganz klar
thank you
rausgekommen,
very
dass der
much
Blick unter die
for
Erde sozusagen, also
this
unter die
very
Oberfläche interessant
interesting
ist.
presentation. I think that's
unserem
very
aktuellen
interesting to
Thema hier im
take a
Hause
look
zum
under the
Thema
soil,
Wurzeln,
also
sich auf der einen Seite
coping
auf die
quite
Wurzeln
well
besinnen,
with
auf der
our thing,
anderen
because
Seite natürlich
roots,
auch sich
thinking of the
anzuschauen,
roots, and
wo
on the other
kommen
side also have a
Leistungspotenziale für die Pflanze her,
close
wo
look
stecken vielleicht auch
where the
Reserven, da
highest potential
weiterzukommen.
can be found
der
in the
Stelle nochmal
plant,
vielen
where
Dank.
might be found reserves. So
Wir
thank you very much
haben
for
auch
this.
natürlich ein paar Fragen bekommen aus dem Chat an
Of
Sie.
course,
Von
there
dem
are
her
some
freue ich mich, dass Sie
questions
jetzt live
coming
zu
in
uns hier im
from the
Studio
chat.
geschaltet sind, damit wir das noch
So
durchgehen
therefore,
können
I'm
und
very
final
happy
klären
that
können.
you came into the studio
So, die erste
live, or
Frage
that you're connected
geht
live to the
zum
studio,
Thema
sorry.
Gerste.
The
Sie haben
first
gezeigt,
question
dass eine sehr
is
stark
about
bestockte
barley,
Gerstenpflanze
because
mehr
you said
Nodalwurzeln
that a
bildet.
very
Und da war
good
natürlich die Frage, die aufgekommen
plant has more
ist dadurch auch die Wasser- und
roots. The
Nährstoffaufnahme
question
verbessert?
is whether with this,
Ja,
if
also
this
im
also
Prinzip schon.
improves the
Zumindest wenn man
intake
sich
of
jetzt
water
den
and nutrients.
Oberboden
Well,
betrachtet.
basically,
Weil die
this is
neuen
the case,
Nodalwurzeln, die
especially if you look
starten
at the
dann meistens mit
upper
einem etwas flacheren
areas of the
Wurzelwinkel
soil, because
und breiten
usually
sich
they
somit
have a
stärker im
very
Oberboden
shallow
aus.
angle of the root, and
Das heißt also,
therefore
wenn dort
they
Wasser-
distribute
und
more in the upper
Nährstoffangebot
layers.
hoch
So
ist,
if
dann
there
tragen die
is
auch deutlich
a
mehr
good
zur
offer of
Wasser- und
nutrients,
Nährstoffaufnahme bei.
then
Einfach
they
deshalb, weil
have
sie
a
ein
higher
neues,
intake of
größeres
water
Bodenvolumen
and
erschließen.
nutrients, because
Nur die
they
ersten
have
Nodalwurzeln, die
higher
schaffen es
volume of
eigentlich
soil.
dann auch in
Only the
der Tiefe
first
im
nodal roots
seminalen, also die
actually
zuerst gebildeten
get
Wurzeln
to
zu überwinden.
to
Aber die
the
später
deeper
gebildeten
areas, the similar ones. But the
Nodalwurzeln, die
ones that
schaffen
are
das noch
formed a
nicht.
little bit later,
Wenn
they
Sie von flach
actually won't do
sprechen,
that.
was strebt
If
man
you
da
talk
oder was
about
ist
shallow,
für ein
what do you
Bereich
mean?
da
What do you
definiert? Sind es die
understand?
ersten 5,
What
die ersten
area
10
is
Zentimeter?
defined as that? Is it the
Ja, nun
first
ja,
5
also ich
centimeters, first
dachte jetzt schon
10
an die
centimeters?
ersten 20 bis
Actually, I
30
was
Zentimeter.
thinking about the first, let's
Ja,
say
noch eine
20
zweite
to 30
Frage zur
centimeters of depth.
Wurzelreaktion bei
Second
einem stark
question, root
überhöhten
reaction.
Phosphorangebot. Gibt
If
es
there
dort
is
eine Wurzelreaktion? Wenn
a
ja,
high
welche?
level of phosphorus, is there a
Also
reaction
noch,
of the root
generell
which
ist
could
es
be
so, dass
found
wenn
there?
das das
Generally,
Phosphatangebot
um,
über den Bedarf,
if
den
the
Bedarf
phosphate
übersteigt,
offer is
dann geht das Wurzelwachstum zurück.
higher
Also
than needed,
Seitenwurzeln wie auch
then
die
the root
Primärwurzeln.
growth
Aber wenn
will be
das
reduced. Side roots and
Phosphatangebot
main roots will be
suboptimal
reduced.
ist,
But the
dann reagiert die
phosphate is
Pflanze ähnlich wie beim
suboptimal. The
Stickstoffmangel,
plant
nämlich
will
mit dem vor allem
react
längeren,
very
vor allem
differently than in the deficiency
Seitenwurzwachstum.
of
Das
nitrogen,
ist dann
which
besonders stark
will lead to a
ausgeprägt, wenn das
longer root
Phosphatangebot lokal ist, wie zum
formation,
Beispiel,
especially
wenn
to the
es so im
sides. And this is
DAP-Bereich
especially
gibt.
the case. This is a very
Dann
local
sind auch
situation.
die Wurzeln, die dieses Band erreichen,
Then the
die haben in der
roots
Regel
that
auch eine
get
etwas
to
längere
this area
Lebenszeit.
also have a longer
Sie haben ja
root.
in Ihrem Vortrag auf das Thema Züchtung hingearbeitet und was man für Chancen
You
auch hier
were
sieht in
talking
der
a lot
Züchtung.
about
Jetzt stehen wir
breeding and
unmittelbar
what chances can
davor,
be found in new
eine neue
crossings and
Regierung in Deutschland
breedings.
zu
Now
bekommen. Denken
we're
Sie, dass man mit
just getting a
Züchtungsmethoden,
new
CRISPR-Cas zum Beispiel ist hier ein Stichwort,
a
da
new
wieder eine
government
neue
in Germany. What do
Diskussion
you
beginnen
think
kann?
that
Oder wie ist da
with
Ihre
this
Einschätzung?
we will start a
Ja,
new
genau das hoffe
discussion
ich
about breedings,
natürlich.
or what?
Also es ist
Yeah,
ja
well,
so,
of
dass
course, I'm
der
hoping for
Europäische
that, of course,
Gerichtshof mit seinem
because this
Urteil
is new,
die
the
CRISPR-Cas, also
European
über
Court of Justice
genomeditierte Editierung
was
veränderten
talking about
Pflanzen, dass man die als
genetic
genetisch
modified
modifizierte
planting, which
Organismen
are
einstufen muss. Und damit
genetic
ist der
modified
Anbau
organisms,
nicht
and
möglich in
therefore
unserer
we
EU und vor allem
cannot
auch in
plant
Deutschland.
them.
Ähm, Der
Affecting
Gesetzgeber hat
the EU
allerdings
and of
mit
course
diesem Urteil
Germany,
auch einen
but of
Auftrag an
course
die
with
nationalen
this
Gesetzgeber gegeben, nämlich
legislation
dieses Gesetz
also
nochmal zu
gave an order
überarbeiten in
to
den
the
einzelnen
national
Ländern. Dazu haben sich
legislations
die
in order
Parteien
to
vor
have
der
a
Wahl
closer
auch
look at it in each
positioniert.
country.
Also zum
This is something
Beispiel
where the
CDU,
different parties
FDP
also took a
waren
position to.
deutlich dafür.
Let's
Bei der
I
SPD,
say the
die waren
SPD
dagegen, also dass dann drin,
is
dass sie dem
against it.
Vorsorgeprinzip
They
folgen.
say
Und bei den
that they
Grünen,
have
da
to follow the
habe ich das
principle
zumindest so
of
wahrgenommen, dass es einen
precaution, and the Green
Umdenkungsprozess
Party,
gibt.
I think
Also
they're
eher die
kind of
ältere Generation
changing
der
their
Grünen
attitude.
dagegen, die
Let's
junge
say
Generation
the
dagegen
older
sehr
ones of
aufgeschlossen.
the
Also das
Green
war zumindest
Party are a
meine
little bit against it,
Wahrnehmung.
and the young
Aber
ones
wichtig ist ja auch, was
for,
die
are
Wissenschaft
in
dazu
favor.
gesagt
That
hat
was
und
at
die
least
Nationale
my perception.
Akademie
But
der Wissenschaften,
the
die
important thing
Leopoldina,
is
hat eine ganz klare
what
Stellungnahme abgegeben
the
und gesagt, dass
scientifics
diese
said
genomeditierten
to it,
Pflanzen,
and
dass die
they
nicht
said that
gefährlicher sind.
the
Und dass wir dadurch
genomic
kein höheres Risiko
edited
durch
plants
deren
are not
Anbau
dangerous, and there is not a
erleben,
higher
als durch
risk
konventionell
when we
gezüchtete Pflanzen.
plant them
Und
than when
da
we use
an diesem
conventional
Urteil
plants.
oder an dieser
And
Einschätzung
this
sollte man sich eigentlich orientieren.
this
Also
estimation is
deshalb hoffe ich
something
sehr,
where
dass
we
das
should
jetzt von der
be
neuen
oriented, and
Regierung
therefore I
nochmal
really
neu
hope that
überarbeitet
the
und
new
angepasst wird.
government will have a
Um auch
new
bei
look at this
Züchtungsverfahren dann
topic too.
dementsprechend
Okay, and when you
zeitliche
talk
Abstände
about breeding
verkürzen zu können.
procedures, of
Genau,
course, in
ganz
order
genau
to
darum geht's.
reduce the
Ich
time
habe noch eine Frage
frame.
bekommen
Yes,
aus
of
dem
course.
Chat und zwar
There's yet
haben Sie
one other
aufgezeigt,
question
dass
coming from the chat.
unterschiedliche
So
Gerstenpflanzen, also unterschiedliche Genotypen
different
bei Stickstoffmangel ein
genotypes of
unterschiedlich
barley
starkes
with
Wurzelsystem ausbilden. Die
deficiency of
einen eher
nitrogen
schwaches,
can
aber
form
waren
a
auch welche dabei, die
different
ein sehr starkes
root
ausgebildet
system.
haben.
One are
Sie haben dann im
weakest and
Verlauf
the
auch nochmal
others are very
gezeigt, dass das
strong.
Gen,
And
das
then you
dafür
also showed
verantwortlich
the
ist, isoliert wurde
gene
und
responsible for that you could isolate that, and therefore we would have the chance in order to use this information information.
Praxis zu
So
bringen.
how long theoretically
Also
would
wenn
be
man
the
das
way in order to
konventionell
put
macht,
that into
dann
practice?
höre ich von
If you do
sich
it
dann in der Regel
conventionally,
zur Zeitspanne von 10
actually
Jahren.
for a
Und
breeder, we're
konventionell
talking
würde
about
erstmal heißen,
10
dass man
years.
auch
And
die richtigen
conventionally would
Genotypen
mean in the
gefunden hat,
first
die
line
man
that
kreuzen
we have
muss.
found the
Das
correct
heißt also, man
genotypes
braucht
that have to be
kontrastierende
crossed in.
Genotypen,
So
die
we
dieser
need
einen
contrasting genotypes
Veränderungen in zum Beispiel diesem
in
Gen
order
BSH3, was
to
da als Beispiel gezeigt
achieve
hat.
this
Das ist zum Beispiel jetzt bei der Gerste nicht der Fall.
this
Da haben wir
modification,
bisher in
like it
unseren
was K3,
Sequenzen
what I
nicht
said.
diese
The case of barley
Modifikation
is not
gefunden.
so. We
Drum
do
bleibt
still
jetzt
have not
für
found this
bei diesem Beispiel
modification
nur die
in
Genomeditierung.
order
Aber wenn man es durch Züchtung machen
to
würde,
use
also
that.
zum Beispiel bei
But
dem
if we
anderen
made
Beispiel, das ich
such
gezeigt habe, da gibt es in der
breeds,
Regel, oder da gibt
as
es
I
auch
already mentioned in that other
unterschiedliche Allele,
example, there
dann
are of
ist
course
das Wichtigste,
different
dass man eben die
attitudes and
Marker hat
different
und dann in
possibilities,
den Nachkommen
and we
immer
would
wieder
use a
verfolgt,
marker
in
and
welcher
then
Pflanze
always
beziehungsweise
in
in
the
welcher Linie aus den
following
Nachkommen
generations,
dann
always look in
die
which plant or in which
förderliche
generation we can find
Genvariante
it
vorhanden
and
ist und die dann
find
letztendlich für
the
die Elite-Linie
promoting
reichen.
genetic
Also konventionell
composition.
Bereich
So in
10,
a
mindestens 10
conventional
Jahre
way, at least
über
10 years,
Modifikation, über
and if we do
Genomeditierung
it via
von 2 bis 3
modifications,
Jahren.
and it would be
Und das zeigt ja
2 to 3
hier
years.
nochmal den
Okay.
enormen Fortschritt, den wir dadurch
So
erreichen
that was
könnten, oder
really
die enorme
an
Geschwindigkeit,
enormous
die wir erreichen
advantage
könnten. Herr
and
Dr. von Wirén,
enormous
wir sind an
speed
der
that could
Stelle
reach
leider
that.
schon
Dr. von
zeitlich am
Wirén,
Ende unserer
so
Diskussionsrunde
unfortunately
angekommen.
we
Sie
already
sehen,
came to
die
the end
Fragen
of
sind
our
zahlreich
time. You
gewesen.
see, the
Deswegen denke ich
questions
auch, das
were
Thema
really
war für
interesting
unsere
and there were
Zuschauer und
many
Zuhörer sehr,
questions,
sehr
and
interessant.
therefore I
Haben wir, denke
would
ich, in Schwarze getroffen.
consider
Vielen
that this
Dank an
topic
der
was
Stelle
really,
nochmal
really
für die
very
Bereitschaft
interesting for our
teilzunehmen und für den
auditors.
breit
So thank you
aufgestellten
very
Input
much
von
for
Ihnen.
taking part and
Vielen Dank, es hat mir
thank you
große
very
Freude
much for
gemacht.
this very, very good
Wunderschön.
presentation. Thank you very much. I really enjoyed it.